5-star 15. So what's her phenotype?
Low function.
Low function, right? And you realize I'm not saying metabolizer, because this gene encodes for a transporter gene and not an enzyme. Transporters have functions, enzymes metabolize. So we call them low function as opposed to low or poor metabolizer. Okay, so patient is, the answer is D, it's low function. Okay, the next question, which of the following are the patient's risk factors for statin-induced myopathy? I think our sister started, but what's the answer to this one? E, all of the above. So what is the best course of action for this patient?
Switching therapy.
D. I'm really working with pharmacists in this one. Yeah, so sometimes it's not just the genetics, and this is one thing that I really wanted to get through with this patient case, because the genetics happens to be just one additional tool that you as a pharmacist, you're equipping yourself with. That's why we believe that pharmacists should be leaders in this field, because it's not just a black and white that, you know, because she's this, don't use this medication. There's other things that are going on with the patient case. You as a pharmacist should be able to ascertain which medications are contributing, which other clinical factors are contributing to this, and be able to help alleviate those and not just the genetics, right? Yes, of course, you should switch the statin to another statin, but is that the only thing that's going on here? What are some of the other modifiable factors that we can actually do something about to optimize the patient's care? So the answer is D. Okay, so we're gonna move on, if there are no questions, to a different case. This is with codeine and CYP2D6. Okay. So coding's story is slightly different. So with some of the other examples, you see a lot of big papers that came out or clinical studies that came out. But with coding, the thing that really pushed it over the edge was case reports. And I just wanted to introduce you to just 3 of those case reports. First, this one was reported, and most of them were in Canada, so maybe Akria might know about this. But this first one was in a 2-year-old boy. He had just undergone an elective adenotonsillectomy, discharged on codeine and acetaminophen for pain, and the patient's parents found him dead on the 3rd day. And the patient post-mortem analysis found out that the morphine levels in this patient was 32 nanograms per ml, and normally should have been somewhere around 0.5.
Wow.
So they genotyped the patient, 2-year-old boy, and he was an ultra rapid metabolizer of CYP2D6. Okay, and so the next patient, 4-year-old boy, also adenotonsillectomy, had obstructive sleep apnea syndrome, and recurrent tonsillitis. So he underwent this surgery to help correct that. Patient was discharged on codeine. This was suspension. Parents found the patient sedated and lethargic after just 4 doses at home. And they rushed the patient to the hospital, but unfortunately he died upon presentation. And the cause of death was ruled out to be bilateral acute bronchopneumonia as a consequence of codeine and morphine toxicity. This patient was also found to be an ultra-rapid metabolizer for CYP2D6. And lastly, there was a full-term infant that just died 13 days after delivery. Normal, healthy baby, died 13 days after delivery. And this case is interesting because the mom was prescribed codeine with acetaminophen for pain, for C-section.
Wow.
And so as she's taking this, she's an ultra-rapid metabolizer, breastfeeding her child, and doesn't know that she's actually transferring morphine through the breast milk to the baby. So the baby died as a result of morphine toxicity as well. And the morphine concentrations were 70 nanograms per ml in this baby. Very, very unfortunate cases. Very, very things that we could have prevented had we done something about. And maybe we don't see a lot of these cases because they tend to be one every now and then. But if this happens to be your child, your mother, your son, your brother, this means everything to you, right? It might be just 1% of the population, but if it's you, it's important. And this led the FDA to actually, this was in 2013. I remember I was doing PGY2 and it came out, I believe, in August, and the FDA said, it's contraindicated. Codeine is contraindicated in pediatrics after tonsillectomies, after adenotonsillectomies and tonsillitis surgeries. And so this is one of the reasons why that came about. And so these case reports show us the importance of genetics, especially for things that codeine is considered a safe drug. I mean, honestly. How many people do you know that have died from codeine overdose, right? It's considered a safe drug, but if it's happening like this and we know the reason for it, we can do something about. So why are we even concerned with codeine and 2D6? Majority of the codeine that's ingested is metabolized to inactive metabolites. About 85% of the codeine that's ingested is metabolized into inactive metabolites. And so those metabolites, like no-codeine or codeine 6-glucuronide, have no analgesic properties. But up to about 15% of codeine is metabolized through CYP2D6, and that gives us the active metabolite of codeine, which is morphine. Codeine by itself is not an analgesic agent. It's as a parent compound, it doesn't give you any pain relief. It needs to be bioactivated into morphine in the body for the analgesic properties. By itself, codeine is an antitussive agent. So yes, you can get that cough relief from it, but it needs to be activated into morphine. And morphine has both antitussive and analgesic properties. And so morphine is the direct result of the 2D6 pathway with codeine. And so with morphine, you can have morphine 3-glucuronide, which has no analgesic property, but morphine 6-glucuronide also has analgesic properties. So this is why we are concerned with CYP2D6 polymorphisms in these patients. CYP2D6 tends to be one of the most clinically relevant genes that encodes the CYP2D6 enzyme. So when it comes to the cytochrome P450 enzymes, the genes that encode them have carried the same name. So sometimes in literature, you see the name italicized, and when it's italicized, it's in reference to the gene and not the enzyme. So it's because they carry the same name. It metabolizes about 1 in 4 of all commonly prescribed medications. So 2D6 is important. If you need to know any gene in pharmacogenomics, 2D6 is in. It tends to be also the most complicated one, the most difficult one to also interrogate in patients, because there are whole gene deletions, there are whole gene duplications, and there are also the single nucleotide polymorphisms, which is just a single base pair change here and there. Interestingly, the very first human genome that was ever sequenced, that individual had a gene deletion, so we didn't know that 2D6 existed until other ones were done. And so 2D6 is a very, very important one. There are over 105 alleles that have been reported in literature. So mind you, there are so many of them. And I want to talk about 2D6 because I think it's, like I said, it's clinically relevant. It tends to be also one of the most challenging ones to understand and to use in practice. And this is why there are over 100 alleles that are known, right? But with these alleles, what's interesting is there's increased function ones. So I said that 2D6 can also be duplicated in an individual. So most people have 2 copies of a gene. You have one from your mom, one from your dad. With 2D6, there can be polymorphisms where you can have multiple copies, more than 2. So some individuals carry 3 copies, 4 copies, 5 copies, up to about 13 have been reported in literature. And so with the increased function ones, you have to have 2 copies or more than 2 copies of normal function allele. So that's how sometimes you see these reported as star 1, star 2 times n, or times 3 or times 4, meaning that it has been duplicated. That particular allele has been duplicated. Also, the normal function ones tend to be star 1, star 2, star 27, star 33, all these other ones that are listed here. Also decreased function 1, star 9, star 10, star 14, 17, 29, and others. No function, star 3, star 4, star 5, star 6. So these ones that are no function alleles can also be duplicated. So if you have a star 4 that's duplicated, it has no function, so it doesn't add on additional ones, right? And so with 2D6, there's a system that we come up with for the naming of the phenotypes, and we call it the activity scoring system. So each one of these alleles have been given an activity value. So those that are decreased function, we give them a value of 0.5. Those that are normal, we give them a value of 1. Those that are no function gets a 0. And so based on the duplication, you multiply the function of this by the number of the duplications as well. So for example, someone might be a star 1, star 17. What does that mean? So if you look in this chart, a star 1 is a normal function in the third row there. So that has 1 activity value, and a star 17 happens to be in the decreased function category here, so that has an activity value of 0.5. So the individual's total value for 2D6 is a 1.5, right? Makes sense. Okay. So these phenotypes are assigned based on the activity score of the 2D6 genotype. So based on the activity score, so if you have more than 2 as your total activity score, you're ultra-rapid metabolizer. If you have between 1 and 2, you're normal. If you have only 0.5, you're intermediate. And if you have 0, you're a poor metabolizer. And that's the different prevalences across different race groups here. Okay. So I wanted to also look at what the CPIC guidelines recommend for 2D6 encoding. It's most important for ultra-rapids and poor metabolizers. Why do I say ultra-rapid? So we saw in the case reports, those were mostly all ultra-rapid metabolizers. They are, instead of just making 15% of the ingested codeine into morphine, ultra-rapids make way more than the 15% of the codeine goes through that 2D6 pathway. So they make too much of morphine, so they should not use codeine as according to the CPIC guideline. They should use another analgesic that does not go through the 2D6 pathway. That's very, very important because most of the opioids are 2D6 substrates. Tramadol, codeine, oxycodone, hydrocodone, they're all 2D6 substrates. Also, poor metabolizers should not use codeine because, maybe I'll let someone else tell me why. Why shouldn't poor metabolizers use codeine?
Codeine is a prodrug.
Okay, it's a prodrug.
So you will not be getting any effect.
Exactly. So you are not converting it into this active metabolite, so there will be no pain relief. And so that's why some people will tell you, oh, codeine doesn't work for me, right? And they want, or sometimes they just want oxy. Oh, party. So they'll tell you that. But it could be legitimate, right? It could be a genetic reason for this. And I think this is actually one of the reasons why this is a public health issue. But some patients might come in and say it doesn't work, and you might just think they're seeking for medication, and they can have a legitimate reason why it's not working for them. So I want us to take a look at a patient case involving codeine. So OD, he's a 21-year-old. He comes to your ambulatory pain clinic complaining of unrelieved back pain, right? He had recently had a car accident. He's taking Tylenol number 3. The physician on service fears that he's abusing the opioid. You remember your recent lecture, pain management pharmacogenomics, and you recommend 2D6 testing to the physician. So after several days, the results come in, and the patient's genotype is star 3, star 6 times 2. Okay, so I'm just gonna go back. Star 3, star 6 times 2. So just think about it here. Star 3, star 6 times 2.
No function?
Poor metabolizer.
Okay, more people. So the activity score is 0. Is this true or false? I just gave you the answer.
Right.
It's true, right? Because if you look at star 3, star 6 times 2, what it actually looks like is that the individual is star 3, star 6, star 6, right? And so you look at the individual activity value of each allele and then you combine it. And so this one is a 0, 0, 0, so the patient is it's a 0. And the predicted phenotype for this genotype is, I think someone said it before. Someone got it? Poor metabolizer, right? So it's D. So what is the implication of this genotype on coding therapy? I think I tried to trick you, sorry.
Okay.
I think it's A. Yeah. Okay, so you get the concept of why this is relevant. So of course you cannot learn all the different examples and the implications for other medications here, but I just chose these two to highlight some important issues that as pharmacists that we can help to add to our medical teams wherever we are. But to be able to do this for others, I want us to take a look at some of the resources that are out there. So the first one I wanted to introduce to you is the FDA table of biomarkers. I know there are FDA workers in the room, so you guys correct me if I'm wrong, but there is this table of biomarkers. You can easily Google this table of biomarkers, it pops up. And it's a list of all the FDA approved drugs that have these sort of genetic biomarkers in their label. And it simply looks like this. I did a screenshot there. What are the content of it is mostly for pharmacogenomic meds and ones that have genomic information in the label, medication names, the generic names, therapeutic areas, what the biomarker actually is, and what FDA, the section in the FDA label that you can find that information. Why this is good is it's a good quick lookup. If you just want to know what is the drug-drug interaction or what's the gene associated with this medication, you can quickly look it up on that table. It's mostly updated frequently, it's publicly available, you don't have to pay for this. However, it's not detailed. It's a very broad list. It needs very it needs review because they're a mixture of both actionable and informative examples. So actionable are ones that you can use in practice now. Informative are things that you know there's an association, but we don't know enough to actually bring it into practice. And so the next one I wanted to introduce is the pharmacogenomics knowledge base. We call it PharmGKB. So PharmGKB is a very, very good resource. You can query medications. This resource can be found at www.pharmgkb.org. It's funded by the NIH, NAGRI, and Stanford University, St. Jude's. They all collaborate on this project. It mostly contains medication and gene search. So if you're looking for any information on medications or the gene itself, this is the good place to look. It also houses the CPIC guidelines. There's relevant FDA information that it pulls here. It also houses information from Japan, Canada, and Europe's pharmacogenomic guidelines as well. Something I didn't write here, but it's a very good resource for those things. So if you looked up a medication and it had a guidance from Europe, it would list it. If it had a guidance from Japan, it would list it, in Canada as well. Also the pathways, the implementation resources, the status of the PGX research, if it's clinically actionable now, or if it's not, it has all of that information. So it's a one-stop shop for pharmacogenomic information. It has a very detailed knowledge base on these pharmacogenetic medications. It could be also very interesting to navigate. You have to get familiar with the website. So the more you use it, the more you know where to look for stuff. But as a person who is naive to the page, it might feel a bit cumbersome to navigate, but it will eventually get better. Because it's so detailed, you have to just know where you're looking. It has a lot of technical details as well, so you have to try to familiarize yourself with some of the basic terminologies in genetics to understand some of these things. Also, the next resource that I wanted to introduce was CPIC. And CPIC is Clinical Pharmacogenetics Implementation Consortium. Happen to be a member of this group, and it's a guideline group that has representation from over 200 countries and over 200 members, I think 50 countries, over 200 members all around the world, and we meet on a monthly basis to discuss new literature, what's prime time for implementation, writing these guidelines, and coming up with the recommendations for clinical use. And so the content of this is mostly the clinically actionable prescribing information. What do you do if you have this genotype? How do you go about devising an active patient plan for your patients based on their genetics? This is the best place. It can be found at cpicpgx.org, cpicpgx.org, and list all the guidelines there for you. So actionable information is freely accessible. There's a standardized format that all the guidelines come in. So once you're familiar with one guideline, you'll know how to just navigate all the other ones, all the information. For instance, if you're looking for clinical recommendations, they're always table 2 in the guidelines. So every table 2 is the clinical information. Every table 1 is the breakdown of genotype, phenotype, and frequencies. So we sort of kept the standardized format so that it's easy to follow and you can find key information very quickly. It doesn't comment on when a test should be done. I think that's one of the biggest things that we're lacking in this space, in this field, is who should I test? It doesn't comment on that. What we comment on is when you have the test, this is how you should use it. Just to also not go into the politics of this whole thing and the payers, and no one wants to pay for all their beneficiaries, so that was a way that we could go around getting the information out there without having to pull in these politics that are associated with paying for testing. Also, most of the pediatric recommendations are inferred from the adult information because pediatric recommendations, pediatric studies are very lacking for pharmacogenomics, and so most of the time we just infer or extrapolate that information from the adult studies. So there are many other resources, and I listed a few here, some certificate programs if you wanted to get familiar with pharmacogenomics. ACCP has a program, happen to be faculty of that. ASHP has a program, University of Florida, University of Colorado. Drug chain pharmacies have a program as well. California Society of Health System Pharmacists have a program. So these days, even if you Google one, I'm sure you'll find one that's closer to you for a certificate program. There's lots of CE programs. We're doing one today, and you can find one at other places as well. There are implementation resources as well. If you wanted to start doing this in your practice, how do you go about it? These are some good resources for you to look at. Many of these questions that you might have already been answered by some people, so don't just reinvent the wheel. Go use what's already out there and just, tailor it to your practice and use it. So all of these resources are also good for that. So how do you go about implementing this? I believe that pharmacogenomic medications, like I said earlier, they are commonly prescribed. The variants itself are very common. So if I was to genotype everyone in this room I would say I would put my money on at least 85% of us carrying at least one actionable variant. They're very, very common, very, very common. In our practice, we've seen almost about 80% carrying a variant when we tested for three genes. If you test for the dozen genes that most of these pharmacogenetic companies test for, almost 99% of the population carry at least one variant. Why you don't see this being reported more and more is because just carrying the variant is not a deleterious event. You have to have that interacting medication for you to have that adverse event or that inefficacy. So that's why we don't talk about these, but everyone carries them. They're very, very ubiquitous. And so we saw that with the Vanderbilt population as well. There is also robust evidence why you should implement this. There's robust evidence for some examples, maybe not for all, but there are compelling evidence for some examples that we cannot just turn a blind eye. We should be doing something about today. And of course, there are technological advances that have helped us to get to this place, because the completion of the Human Genome Project helped us a lot to get all the tools and also have a reference point for what the reference genotype is. And I always wonder who picked the reference person, because who is the reference? It could be me, it could be you, right? But it's just, we tend to use what is most common in the general population as the reference, and anyone who differs from that tends to be the variant, right? And so also the testing costs that are declining. The first human genome cost over $2 billion US dollars, yes, Trump dollars, to actually sequence one person. And now you can get a sequence for less than $1,000. And so for how far we have come with the decline of these costs, it's very impressive. And especially for genotyping, it's way less than that as well. So we can do this. But if you want to do this, what are some of the challenges that are associated? Of course, there's a huge knowledge gap. There's a huge knowledge gap between the researchers and the practicing clinicians. So how do you bridge that gap? So that's why I believe pharmacists, and that's what the ASHP said and the competency group said, that pharmacists need to be the ones educating their team members. You know this is just one tool. Just as there was a point where pharmacokinetic testing was not necessarily a thing, but now it's like normal practice everywhere. You renally adjust therapies, right, and doses. This is just another way. Just think of it as you're just genetically adjusting your doses and therapies. It's just an additional tool, so we need to take a lead on this. Also, there's a lack of reimbursement for some examples, so it's making it hard. There are some examples that payers will pay for, like C2C19 testing, like, and some antidepressants and antipsychotics they will pay for. So some cancer therapies they will pay for as well. So not all these examples are being paid for, but there's a lot of progress and work in ongoing now that is hoping to answer that question and get back to the payers. So we did have a payer engagement meeting about a year ago in DC, and just getting all the payers around the table and asking them, what will it take you to pay for pharmacogenomic testing? And when they told us what they would like, the group is going back and just coming up with a study that will address all those concerns, and hopefully they cannot say no when the results come out. So there's a lot of work ongoing now that's hoping to address that. And there's a lack of infrastructure. Not every institution might have electronic medical records around the world to do this, or to be able to even have access to the genotyping facilities. And so that might be a challenge, but there are ways to go around it now. You don't have to do it in your institution. There are send out tests that you can request and other things. And also clinician resistance. I think most people are just resisting it because they don't know enough about it. So some of our primary care providers would say, well, if I get the test and it doesn't only apply to medications that I prescribe, then what do I do about that, right? Am I liable to the results? Am I liable if something happens? And so we have to find a way to these are all logistical things and things that we can work through at various institutions, and find a way to not just get the information to that one provider, but every provider that's involved in the care of the patient. And also some of these evidence is evolving over the years for some examples. So to do this, there's a planning phase, there's an implementation phase, there's a post-implementation phase. I want to talk first about the planning phase. What is your motivation? Why are you even doing it if you wanted to start one, right? So why do you want to start a program? Are there any particular drug gene examples? If you work in psych, there are so many examples there to start. If you're probably in a cardiology clinic, clopidogrel is a good example, especially if you have Asian patients. because they tend to carry the loss of function variants. So maybe you have different motivation to do it. Do you have the target population? Do you have the right medications you're starting with? You don't have to start with a whole list of meds, maybe just one example. You're just, testing them for one thing and just guiding one therapy that you prescribe a lot in your practice. Also, you gathered some information to also help you make a case for this. In the past, how many patients have been prescribed clopidogrel and had an instant thrombosis or came back with another MI or stroke? Maybe if you have this data, you can say, we should be doing something about it. Show where there is a lack in the institution and hopefully get some support for it. Also, what are the resource needs? If you wanted to do this, who are the people you need? What are the resources, the infrastructure that you need? And then consider that. And if you do have some of this in your institution, great. If not, how can you outsource that resource need? And also, what type of program do you want? Is it just a research project, a quality improvement project, a clinical rollout, and change in practice? So these are all some of the things that you can consider. But with the implementation, you definitely need an oversight committee. Ours consists of a multidisciplinary team. I lead that team with there are MDs on there. There are lab technicians, lab experts, geneticists, research coordinators, admin support, and of course institutional leadership as well. And also, you review that evidence in that group because you want that group to actually be the ones that give you their buy-in first. Do they agree? Because the physician champions are going back to the physicians and saying, this is what we have to do. Our patients need this. Sometimes it's better when it's coming from a peer rather than a pharmacist going into the practice and saying, you need to do this. They're like, who are you again, right? And what do you know about my line of work to tell me what I need to do? And so you need the other people, champions from different disciplines, even nurses, especially if you're getting blood draws and things, you need nurses on board because they can facilitate that sample collection process for you. And design your workflow, designing these protocols and support tools. All these things, the workflow, the support tools, even some of the evidence are already done for some examples with the resources that I showed you. So if you're ever interested, that's a good place to start. Some of these things have already been done. You can take that information and just implement it in yours. But even after implementation, there is a need to keep doing that collections, data collection. Are you actually improving the care? Are the doctors using this information? How do the patients feel about this? What type of patients are you studying? Is it Hispanic patients, African Americans? We need a lot of those studies in our populations. So this is important. There's education, there's also maybe implementing new ones. After you successfully implement one example, you can go ahead and implement new ones. Okay, so just enough about the theoretical part. I just wanted to quickly show you what we do at Mount Sinai as well. Except that my slides are not moving. Okay, here. So our team, we have a lab that's translational pharmacogenomics, translational initiatives in pharmacogenomics, and it comprises of a group of people. This is definitely not a one-person job. You need a whole team. We have Semaphore, which is our genetic testing laboratory. It's a company that's been spun out of Sinai. I'm part of the Charles Bronfman Institute for Personalized Medicine, also part of the Department of Genetics and Genomic Sciences, and of course there is also the ICON Institute of Genomics and Multi-Skill Biology. And we cannot do this without our clinical partners, so we have a lot of partners around the institution, mostly in our outpatient clinics, our primary care, our faculty practice, and associate practices there. But for all of this, we have three main aims. First is our educational aim. We intend to educate our patients. We intend to educate our providers. And also for our implementation, we have clinical ones. We have ongoing ones like IPM PGX and eMERGE PGX, which is funded by the NIH and NHGRI. And also we just trying to implement across the entire health system. This is something that's actively going on right now. And hopefully we can do this for all employees of the health system. In translational research, developing evidence to support some of the ones that we don't have enough evidence for. So far with our outpatient efforts, we've enrolled about 1,600 patients. 420 of our physicians have signed up to be part of this program. Very, very diverse group of